ऑन कर दीजिए,
रिकॉर्डिंग ऑन कर दीजिए।
हाँ हाँ, आई कैन सी रिकॉर्ड।
From there it was. From there it was. From there it was.
आप कर रहे हैं रिकॉर्ड? Streaming has begun?
Yeah, it's better.
Okay.
Now you can start.
I can see record.
We have to feel screen. From there it was. From there it was. From there it was.
Good morning, everyone. Thank you for joining us for this new series of talks that we have, which is called Futuristic Fridays.
Streaming has begun.
And in Futuristic Fridays, we are going to bring you a series of 4 talks that will talk about the interface of AI in biology and health. And now we thought that this is futuristic for multiple number of things. One is AI is everywhere. Good morning, everyone. Like, thank you for joining us. Newspaper you open, what magazine series of talks that we have, which is called the Futuristic Fridays. And in Futuristic Fridays, however, we are going to bring some new buzz around a series of 4 talks. It's also important to remember that it's not like it has come interface of AI in biology. For decades and together, with multiple number of people have worked. One is AI is understanding everyone. Like, thank you for computational method you open what series of talks AI is like, which is called using these methods. And in Futuristic Fridays, however, we are going to bring some new buzz around a series of 4 talks are one to remember that areas that's not like it has come about of AI in biology and in these and now in this series multiple number of people have worked. One is AI is understanding everyone, like starting today no matter computational method you will be talking about AI is like which is called using these methods and in Futuristic many however we are even of course some new buzz around studies of engineers are people who are key areas that's not like it has come about of AI in biology and in these and now in this series multiple number of now will bring the 4 one is AI in understanding everyone like starting today no matter computational method you will be talking glimpse of AI is like which is called using these methods using many however we are of course some new buzz around studies of engineers are people who are key areas that's not like it has come about of AI we will also be having and now in this series now will bring the 4 one is AI in understanding everyone like starting no matter computational method glimpse of AI is what is called using now using many however we are of course studies of engineers are people who are key areas that's not like it has come about of AI we will also be having and now in this series now will bring the 4 one is AI in understanding everyone like also stay to have the ability to go beyond just listening to lectures, that you also participate in solving some of the questions with these experts. So if you register on this, you will get to know about all the other future things that we have incoming for the Futuristic Fridays series. In addition to that, I will also take the opportunity to tell you a little bit about the Jigyasa program under which we are doing this series also of Futuristic Fridays. Jigyasa is the flagship program from CSIR that connects scientists with students who are in schools who are still thinking of joining science, or maybe they might not join science, maybe they go on to do humanities. But we are living in an era where knowing about science and technology is a necessity for everyone. So starting from school students to college students who are already pursuing science, who have already taken the decision of getting into the depths of science. So Jigyasa really is trying to bridge that gap of students and real life science and the scientists that work in the labs such as CCMB, where some of you are sitting here today. So Jigyasa is trying to bridge that connect. And we do that by various different methods. On this website, Jigyasa, you will find various different kind of digital content. Sometimes, we all want our students to be doing hands-on experiments, but that is not always possible. And hence the community has spent a lot of time developing content where you can do digital simulations, digital experimentation, in addition to having a lot of resources that teachers, for example, can use in their classrooms. Ranging from these digital content to a lot of in-person events, Jigyasa offers all of this. And we really would appreciate if the education community, students as well as teachers, really take advantage of this. With that introduction to Jigyasa and to the Futuristic Fridays talk series, I will now invite upon the speaker for the day, Dr. Arvind Rao. He joins us from University of Michigan, where he works as associate professor. And his expertise is really in engineering and trying to put that in healthcare sector. More details will be from Arvind himself. Arvind, up to you.
Hi, everyone. My name is Arvind. First of all, thank you for inviting me to participate in the first lecture of the series. It's a dream come true for me at several levels. CCMB is a very hallowed institution, so to give a talk in this place is a big thing for me. So thank you for inviting me. It's also very special to talk to students here at from the school system here. I was a product of the school system here. In fact, I grew up in Hyderabad many years ago. I went to a state board school called St. John's School, then I went to a ICSE board school called Gitanjali School in Begumpet. And then it was my life's ambition to go to one of the CBSE schools, including several of them are here. So it's a double pleasure for me to interact with all of you. I'm going to come at this from an engineer's perspective, which is biology, in my mind, is the new physics. What do I mean by that? If you ever have thought about physics, the part that makes physics really interesting is that there's an extremely wonderful instrumentation to understand physical phenomena, which could be from nano or femtoscale or attoscale all the way to macro. You can look at galaxies, and we have lots of data, and you want to be able to interpret this data in the context of known physical laws or discover new physical laws, right? I think biology is basically at that exact point, right? It was at that point about 10 years ago when the human genome got sequenced. And when the human genome got sequenced, there was this promise that now we know every nucleotide in the human genome. What does it do? Every single base, A, T, C, G, it's in some really interesting order. Why is it like that? What does it do? And how do we understand what it does for the organism? How can it be that my DNA resembles the DNA of a fruit fly, the fruit fly that you see, hovering around your fruits at home? We almost intersect about 96% in genomic variation in DNA. Yet we are so different, right? Or me versus a troglodyte, which again 99 point something, right? So different. And socially we have different behavior. We do something similar, even between flies and humans. We do certain things very similarly. But it's amazing that that's the level of overlap in DNA between these two species. And you can do this now for all the species. You can sequence the DNA of so many things. What does it all mean? And we can do it in two ways. Can we use the DNA to understand why something is built a certain way? And if you understood it well enough, you can figure out how to build something new, right? So people are talking about resurrecting the woolly mammoth, which is extinct, by the way, right? But people are saying, okay, somewhere there is some DNA if I figured out how to sequence the DNA of a woolly mammoth, I could basically bring it back to life using modern DNA techniques. So that's the power of DNA and the power of biology. The instrumentation to understand genes, proteins, metabolites in the human body is unprecedented. We have lots of data. We don't know what to do with it. And unfortunately, biology has traditionally been a very non-quantitative discipline. But I think the time is perfect to change. It's time to think about statistics, to think about mathematics, think about computer science to understand biology. So if you, and I'm not going to, go any far beyond my own self. I really enjoyed biology in school, but the part I could not do to save my life was I was not very good at drawing. And my drawings are always the worst in class, right? You could tell this is Arvind's record book because the drawings were almost pathetic. The other part was I'm very bad at memorization. So if you gave me a bunch of facts and said, by heart this and come and put it out in a paper tomorrow, not going to happen. I'm not constructed that way. I'll come back to this in a bit. We're all different kinds of learners, and it's okay to embrace that diversity in the way we learn. I think we all can bring something unique. Okay, but more on that later. So I left biology for the wrong reasons. I didn't know enough of biology to understand that it is actually a very beautiful subject. But it dawned on me only when I went back to get my PhD. And I get back to get a PhD in engineering, not in biology. I didn't even imagine I will go back to biology. But I happened to work in a laboratory where, because a human genome had been sequenced, people said, well, we have lots of data. We have no idea how to analyze it. You're a math guy, right? Come help us understand this data. So I walked into biology accidentally. It was a very lucky and fortuitous situation in my life. And I wish those accidents happened to you too. Because otherwise, there was no way I was going back to biology. And then, to make matters even better, my PhD advisor realized I knew no biology. The good thing was I set the bar really low. So he said I knew no biology. So you know what? Okay, fine. Let's just spend 3 hours in my office. I'll teach you all that you need to know for your project. And in those 3 hours, he gave me a crash course in biology. And I then began to process how beautiful a subject it is and how complex it can be once you understand all the pieces. And to embrace that complexity, you have to think integratively. You have to bring computer science, statistics, data science. Biology is not a qualitative science anymore. I don't think it ever was. It just got the wrong perception. Right? Maybe the only math you've seen in biology is when you did your Punnett square for genetics. There's a lot more to biology that's quantitative that we never talk about until much later. So I want to encourage, if I want to leave one thing with all of you, is I would encourage you to think integratively. If you're picking biology, if you don't want to do biology because you know enough to know that you don't like it, that's fine. But don't make my mistake. I walked away prematurely because of a perception that I then learned to be incorrect. So don't make my mistake. Right? I think there's a beauty to biology that we talk about as we go through. So in my lab, we do a whole bunch of things. We basically look at biology quantitatively across multiple scales. What you see here is essentially DNA profiles for different kinds of cancers. There's also, you can get microscopy images of different cancer cells. You can get radiology images, that's an MRI scan, and you can get pathology images. So what we build is AI methods to be able to bring these together. I'll also say this maybe one time, I might get to this a couple other times, but to me, I don't understand the word AI at all, right? What I mean by that is people say it's artificial intelligence. I don't know what's artificial about artificial intelligence, right? I actually think it's augmented intelligence, right? Because only a human AI, a human machine team can synergize to help do things that neither could do by themselves. So it's augmented intelligence. It's not artificial in that sense, but again, don't quote me on it. All I'll say also is that AI has gotten sort of a strange perception to it, in the sense that it's not new. It's been there since the 1950s at least. Thank you. Thank you. It's been there since the 1950s at least. Middle point, right? Battery. That's okay. That's okay. No problem. Okay, I'll try. I always forget my glasses, so I don't know. Okay. Thank you. So, but AI is not a new field, right? If you read about the history of AI on Wikipedia, you'll come across some names. Marvin Minsky at MIT. These people tried to really build the first engines to understand the brain, right? How does the brain work? How can a few neurons program themselves to be able to process information? How do we deconstruct that? Can we deconstruct it even, right? What's happening systematically? And so there's a lot of statistics and what is now called machine learning that happens in what is called AI. So AI is just a nice name, but it actually means conceptually a lot deeper things than we are used to. So don't get swayed by what you see in the newspapers. Newspapers tend to have a way of trying to obfuscate the history of the subject than what we are used to. How many of you, I know your teachers are here, but you can still see if you feel comfortable saying it. How many of you use Flipkart or Amazon? Okay. How many of you use Netflix? Okay. How many of you use Spotify or one of these Pandora or Savan? Okay, good. So you all use machine learning. You all use AI already. It's already happening in your life. What does it do? You buy a book, it recommends another book for you to buy. You watch a movie, it recommends another movie for you to watch, right? And whether you like it or not, right? The algorithms are grouping you into different categories. Oh, you're all a bunch of 16, 17, 18-year-olds who are in school. So let's just bombard you with ads from Bijus, right? That's happening all the time, right? Or you're in first year college and you just found your freedom. So let's tell you all about, this cool new pizza joint in town, right? They know what they're doing. They're grouping you into different categories based on certain attributes about you, and then using that information to be able to target you in some way, right? And that's all basically machine learning in the scene. So now the question is, machine learning has been used commercially because there's lots of data about us that's on the internet. Can we use machine learning in biological hypothesis and discovery? That's the question. So I'm going to breeze through this in the interest of time, but also to leave you with intuition. I'm not trying to get through to technical aspects. If you care about technical aspects, we can talk about it after the talk if there is time, or you can email me too. But I want to leave you with intuition. So the question that you should always have in your mind is, does this matter? Why should I care? And what's my couple of takeaways, right? The mechanics of what I'm talking about doesn't really matter, okay? And if you have any questions that are really burning and you say, why should I care? Don't hesitate to ask, because this whole conversation is about you, right? So when I come into this is, I was just thinking about, we're talking about nonlinear optics. So I came across this and I said, this is actually a pretty good metaphor. And the metaphor here is one of you must have all seen this in school at some point. This is called the double slit experiment, very classic experiment. It's called a Young's double slit experiment, where what you have is you have a light source, you have two slits, split by a certain distance. I think it's lambda over 2 or whatever. And basically what happens is it splits. And in the end, what happens is there is constructive and destructive interference. And so you get these bright and dark fringes, right? And in my mind, to me, this is a metaphor, right? And the metaphor is, this is biology and this is math, right? And there are lots of places for both constructive and destructive interference. So I'll say in my life, that's what happened. There was a point where I began to see these two things together. And all these interference fringes that you see are places that we found to synergize biology and math. Today, it's come to a point where I can't see biology without math. I can't see math without biology. I think it's an amazing intersection. This is my background. I got my engineering degree in India. I went towards physics and math. I didn't take biology, even though I wanted to be a doctor. I realized I can't memorize stuff and I can't draw to save my life. So I said, medicine is not for me. Bad idea. I should have persisted. So I leave that thought with you is, don't give up on a subject because of some misperceptions. Okay. So I went and got an engineering degree in India. I was in Bangalore at the time. I then went to UT Austin in Texas, got my first master's degree in wireless communications. And I just happened to take a course. Out of all the courses I had to take in my graduate course, I took one class in data mining at the time. It was called machine, it wasn't called AI, it's called data mining. So I took a class in it, and just so happened, the professor said, why don't you try working with data mining on a biological data set? Complete accident. And so I applied some very simple ideas from what I had learned in engineering to biology. And strangely enough, that paper, that result was publish worthy and got published in a conference, something I never expected to do. So then it occurred to me, hey, there's actually a nice pollination between what I can do as an engineer and what problem biology has in store for us. So I then went on to Michigan. I got a few degrees, and then I then went to Carnegie Mellon in Pittsburgh. Did some more, did a postdoc there, and then I went to a place called MD Anderson Cancer Center in Texas, and did my faculty. I was a faculty there for about 7 years. Then I came back to Michigan because basically I love Michigan. The university has been very kind to me, so I came back as faculty 5 years ago. And so far, my group has been working on things like understanding images in biology. How do you make sense of images in biology of different kinds? They could be radiology images, they could be pathology images, they could be cell images from microscopy. And we also do a lot of data integration, which means different things to different people. But what one application is, let's imagine a person whose blood draw is available to you. So you have a small sample of blood from that person. I can now sequence the DNA in the blood, and I can tell you with a fairly reasonable resolution what the status is of all the genes in that person's blood, right? This gives me a sort of a baseline. Now I say this person goes out into the world and contracts some kind of infection. It could be the common cold, it could be the flu, whatever, right? And there's going to be a change in the biology of the person, right? They're going to feel sick. And sickness has a consequence on the expression of the genes in that person, because that's biology, right? Now the question is, how does the genes expression or the genes measurement evolve as a function of the exposure to a pathogen? And what we found at the time is that the measurement taken by these genes is a direct reflection of the pathogen that infects the person. So there's a unique signature, a unique profile of genes that are in a person who's got the common cold versus someone who has the flu. And why is that useful? Because we wanted to do one step further, which is it's possible to get a blood draw from a person, twice a day, or every once every single day. And what we found, which was amazing at the time, if not so surprising anymore, was that you can predict that somebody's got the infection of a certain kind much before they begin to show symptoms. So you might be feeling perfectly fine today, and you are probably going to have cold in 2 days. I can tell you you're going to have the common cold, or somebody else might say, I'm having a bit of a sniffle, I feel a little bit of a headache. I can tell you with high probability whether headache is attributable to the common cold or it's something indicative of something deeper like the flu, much before you begin to even see these symptoms. So the power of biology combined with data science is to be able to do things like prediction, forecast. Just like you can forecast the weather, you can forecast the symptomology of patients with this kind of mathematics. So I'm going to give you a 5-minute primer on data mining, right? What is data? I'm using this word a lot. Just to get everybody on the same page, I'm going to use a few words quite often in this presentation. So let's just say I have a table. The table is on the right. It's got, the first column is simply, the instance, which is, patient number one, fruit number one, fruit number two, fruit number three. So what I've got is a whole bunch of fruits. The fruit name is on the last column. So that's called a response, right? In this kind of tabular data structure, that last column you see is called a response, or it's called a label, right? Imagine your books, right? Okay, fiction, non-fiction, crime theory, whatever it is, right? Like, so every genre of book or movie is a label. And then in the middle, between the first column and the last column, I have this bunch of measurements: the shape of the fruit, the color of the fruit, the size of the fruit. And every fruit has a different set of attributes, right? Apples are red, bananas are green or yellow, depending on which kind, right? So you now have different features that describe a particular fruit. Think about this in terms of books or movies that you like, right? Who acted in the movie, how many characters, Is it a fiction story? Is it not? And then did you like the movie or not? That's exactly what Netflix is doing, by the way. Netflix is building a table for each single person in this room. They have a unique table that says, you watch these movies, the movies had these attributes, and the last label in your case is whether you liked it or didn't like it. If you liked it, it means you watched the movie till the end and then you liked it. Or if you stop watching in the middle, it probably means that you didn't want to watch it further. So it makes all these conclusions about you and tries to predict the label based on the features of the movie or the features. So that's what's happening. Is the intuition clear? Anything that is not clear? Okay, so you have instances, you have labels or responses in the end, you have features in the middle. That's the three things you want to keep in mind. Now, if you wanted to, the typical problem machine learning is to predict the status of this last column from all these features. In your case, it would be, do you like this movie or not like this movie? So when a new movie comes in, right? Like a new movie just got put on Netflix, right? Let's say whatever the new movie is, like Fighter. Okay, just shows up. So, okay, if you like Hrithik Roshan movies and you like Top Gun and those kinds of movies, you probably will like Fighter. If you didn't like Hrithik Roshan or you don't like Top Gun type of movies, then it will say, okay, you don't fit the bill, so you will probably not like it. And why does that matter? Because if you like the movie, it will show it to you and ask you to watch it. If it knows you won't like it, then why bother, right? Show you some other movie that you might like. So this is what is happening in the under the hood. So that is called supervised learning, where I want to predict the label, whether it's an apple or a banana, based on all these features. Right? Same thing, whether you want to watch a movie or not, whether you want to buy a book or not, whether you want to listen to a song or not, you can be thinking of in the exact same framework. Okay. So the second is, let's say I didn't have this label. Let's say I block it for a second, and I say, let me simply try to group the different instances based on these measurements. Right? Thank you. Let me just drop this label. Let me just try to group all the instances based on these measurements. Now, what I'm saying is, I don't tell you what type of fruit it is. I'm just saying, use the fact that it's round, red, normal, or ellipsoidal, green, or long. Simply group them based on these attributes. What do you think might happen? Anybody wants to venture a guess? I don't tell you what fruit type it is, so you don't have this information. Simply grouping based on these features. What do you think might happen? There's no right answer, by the way. So if you can say whatever you feel like.
All the objects having same color will be grouped in one category, and all the objects having same shape might be grouped in one category, and same size will be grouped in another category.
Beautiful, beautiful. So I hope you heard his answer. Depending on the choice of the attribute, you can have different kinds of combinations, right? So if it's based on, and that is called a distance measure, right? Nothing fancy, right? Distance measure says if you're categorizing objects based on their similarity in color, you might have a very different grouping than similarity based on shape. So oranges and apples will go into the same group if you cluster based on shape. If you cluster based on color, they'll go into different categories. The exact same set of objects. So picking the right metric, the right distance choice is critical when you do this kind of stuff. This is called unsupervised learning because there's no supervision. I don't provide this class label. Unlike the previous situation where I gave the class label and I said, predict this based on these features, right? There's no supervision anymore. I'm just doing unsupervised learning by saying, find me all the groups in the data. And then, of course, I've talked about Netflix and Amazon already, right? What it's doing is something called active learning. Active learning basically means it'll show you a movie. If you watch it, it'll say plus. If you don't watch it or you don't like it, it'll say minus. It'll keep using this combination of pluses and minuses to figure out what's the right function that maps to your choice of movies. Okay. This is happening in our daily lives. So now I'm going to take it to biology, right? Which is, what do I want? Biology is in this context, it's about trying to interpret genes that make a cancer cell aggressive. Okay, so to all of you, you know about cancer in general. And we talked about the fact that there are about 20,000 genes in the human genome. And different combinations of these genes can make a cancer cell either very aggressive or not as aggressive. And the reason that's important is if it's very aggressive, it means it'll grow very fast. It's very bad for the patient. It's harder to treat. If it's not as aggressive, it'll grow slower. There are some ways to keep the tumor in control. And maybe, just maybe, it might be a little bit easier to treat. Right? So that's very important to understand how these cells behave as a consequence of their genomic constitution. So we are working with a disease called TNBC, which is called triple negative breast cancer. Triple negative breast cancer is an especially aggressive form of breast cancer. And the reason it's, and because it's triple negative, nothing that's negative can actually be good, right? So triple negative is really, really bad because the typical treatments we use for hormonal therapy of these breast cancers are not available to treat these patients. And the incidence of this is rising. And it's rising especially in areas like our country, in Africa, right? And so this is becoming a very important problem to think about. How do we understand the genomic constitution of these triple negative breast cancer cells? The hope being, if you understand this constitution, maybe, just maybe you can treat them and help these patients. So what we have is a situation like the following. This is a very typical high throughput data set, or, because I just told you that biology is very quantitative. One way where people do these experiments is they have these kinds of plastic plates. So this is obviously a very nice rendering, but in reality, it's a plastic plate that looks something like this, right? And what you have are multiple dots in a grid. It's called a high content well. And in each little well, you can drop cells. So what you're seeing is this kind of view for this kind of dish. Okay, make sense? All right. So now you got these wells, you drop cells into these wells, and now you have different drugs. The idea being, I can now try all the drugs that are known to mankind and ask the question, how do these cells respond as a function of that drug? The hope being that if I find a set of genes or a set of drugs that might work on these cells, and they're all triple negative breast cancer also, hopefully it will help identify some potential drugs that could treat these patients. That's the promise of the problem. Maybe I'll take a quick detour to tell you what is wrong with this assumption of trying to take cells out of a patient, putting them into wells, and then putting drugs to those patients, to those cells, and treating cancer that way. Is it a good idea? Is it not a good idea? What am I missing? How many people think it's not a good idea? How many people think it's a good idea to find drugs that way? Would I encourage somebody who said it's not a good idea to say something about why they feel it's not a good idea? Go ahead. Sit and talk.
Possibly take so many cells and try every combination. Such a manner, it would be very painstaking and time taking also.
Beautiful. So you just talked about this problem in biology, which is that cancer is very complicated for a reason, right? If every cancer cooperated with a single drug, life would be a little bit simpler. But reality is you need a combination of drugs. And what she's saying very rightly is that if you had to look at all drugs and their combinations, that would be more atoms than atoms in the universe, right? So it's not feasible to do it directly. Somebody else had a point. It was the same point or a different point. I'll tell you one more thing. How many people think that we are all two-dimensional creatures versus three-dimensional creatures? Are we two-dimensional creatures? Anybody think we are two-dimensional? Are we three-dimensional people? Okay. And are we homogeneous? Is the cells of the brain the same as the cells in the feet? No, right? So there's a very, very clear architecture that's at play, and the organization of these cells in the different parts of the body has a consequence for what you can do where, right? So because biology is three dimensions, working with two-dimensional constructs is actually not solving the original problem, but it's the best that we can do because you want to be able to cite content. So this can help you filter down some drugs, but is not the whole picture. I want to keep that at the back of your mind. So what we now do is we take these cells, put them into these plates, and in every well, we put a few different drugs. And then we take pictures of them. Now, this doesn't do justice, but I'm showing this to you for a reason. The pictures actually taken from this setup. These are cells that grow in clumps, and I seek your forgiveness because I know it's not very clear. But I'm showing this to you because this is real data. And why am I showing this to you? To tell you that the textbook representation of biology that you see is only illustrative. It's not real. It's an artist's interpretation of the problem. It's not real data. To get to real data that looks that clean, you have to do a lot of work in the background that never gets talked about. So biology, just like any other science, is messy. If you think that biology is going to be as clean as what you see in your textbook, you're mistaken. So data is very messy. You have to be able to clean it up. But here's the intuition, and I think I have a picture for this. The cells look like, how come this didn't change? Okay, there it is. I hope it won't. Okay, let me try again. Okay, so these cells, they grow as clumps, right? Because that's the tendency of cells to want to stick together. But these cells have two morphological differences, right? One is called an epithelial cell, which is sort of a cell that's balled up like this, right? Another is a mesenchymal cell that's very aggressive, that kind of opens its fingers. And why is this? I'm just telling you, this is worse than this. Why might that be? Why is this really bad? Why is this not so bad? In terms of think that you're a cell, why is this making you more aggressive than this? Good. When I have protrusia, think about amoeba, lamellipodia, and all that stuff, remember? Right? It's easy to move. So for mobility, the more protrusions you have, the more the ability of these cells to move. So these mesenchymal cells have a lot of properties, one of which is extreme motility or mobility. And so the idea being that these triple-negative breast cancer cells typically look like this. We want to find those drugs that will make these protrusions go away. So I want to add a drug that will convert this to this. For people on Zoom, I'm showing my open and my closed fingers here, and just saying that if you have, the idea is to find a drug that will cause the recalcitrance of the mesenchymal phenotype to epithelial phenotype. You might ask, well, that's okay. Why not just kill the cell? What's the point of keeping it alive and changing its form? We take you to a different concept that you studied in biology. You all studied ecosystems and habitats. Pick your favorite area. Think about the beauty of the area, right? There is grass, there's tigers, there's goats, there's rivers, there is dry spots and there is wet spots. Take any one of these things out of that and tell me what will happen. Let's take the grass out of the equation. Can this ecosystem survive? Take tigers out of the equation. Will the ecosystem survive? So what am I trying to say? This, correct. Exactly. So the idea is a very, very exquisite balance between all these different species. If they exist, they actually cooperate. If you take any one out of the equation selectively, you're going to create a worse situation. And in tumors, that's called tumor resistance. Tumors are heterogeneous. They have different kinds of cells. If you only kill one kind of cell, the other cells that are not responsive to the drug will begin to grow and proliferate. And next thing you know, you just made a bad problem worse, right? So the idea would be killing the cells is not as difficult. You can add cyanide, right? Really. And that's what they do, in fact. In chemotherapy, that's the kind of drugs they use. They're pretty potent poisons, right? But can we do better? Can we not just killing the cell is not the problem? Can we actually, if we make these cells become less aggressive, you've taken away their legs, they can't move. Now they're all localized. Now you can do surgery. It won't like just run away and start metastasizing and those kinds of things. So that's the idea of trying to find drugs like this. I hope the intuition is clear. So now, talking about data mining, right? Now, what do? I have images. I extract a whole bunch of features from them. I told you about features in the previous slide. I'll come back to it again. I do some data cleaning. Like I told you, real data is messy. You have to clean it up. After cleaning it up, you extract features. You run some data mining models. I'm not talking about AI, very basic data mining models, and then try to find those drugs that might cause the behavior I look for, which is converting this to this. Again, for the people on Teams, I'm talking about the conversion from mesenchymal phenotypes to epithelial phenotypes. And once you do that and you find drugs, then we say, why does the drug work? Right? What's the drug doing in the system that is making it especially work for this sort of problem? So I told you about data cleaning. I'll skip that in interest of time, but it's intuition that it's dirty data. You have to clean it up. And so the features that we typically obtain from this kind of images is the following, right? Like I now go back to my example of fruits, right? Now instead of fruits, I have an image on every single row. And every measurement is how round it is, because you can tell if you think about, again, math, right? You all studied ellipsoids, you studied eccentricity. Remember eccentricity, the E, when E is equal to 1, that's basically a circle. When E is not 1, right? Major axis divided by minor axis, right? It's the lower E's or the more E's, it's ellipsoid. So you can make measurements like the shape, how eccentric it is, what's diameter, what's area, etc. Right? And all these measurements become the features in my table, right? And once I have that, I have now come my big problem. I get about 20,000 images from this experiment. And I need some nice person to come in and label these images so I can build a model. Good luck trying to find somebody who will spend their time and label 20,000 images for you. It might be really nice, but time is always a problem, right? So what do? You say you start small. I just went to my collaborator who knew the biology and said, hey, I know you're really busy. Would you mind? Here's a Cadbury's bar. Here's some coffee. Okay. And can you please spend some time and label some images for me? He said, okay, I have time to be able to label 33 images. So you took 33 images from those 20,000 images and labeled them as either this category or this category. Long projections or no projections. Okay. When I say this, I mean long projections. And when I say this, I mean no projections. So he gave me 33 examples. And I had the data like I showed you for the different kinds of fruits. And I went ahead and built this model. So the model that I built is very, very intuitive and very easy, nothing nonlinear. It's basically called a decision tree. And we all do decision trees in our life. Oh, I need to go and play, but I have an exam tomorrow. Do I have time to go and play? So I make a decision. Oh, actually, I finished 80% of the syllabus. I have about an hour. Maybe I can go and play. Right? You make decisions that are very, very bivariate in your life all the time. Oh, I have a limited appetite, and there is this sweet and this sweet and this ice cream. How do I decide what combination of sweets I can have to be able to satisfy my appetite but not overeat? Right? We're doing this all the time. So this is an example of where you might decide to play, where you have some variables like, okay, what's the weather looking like? Is it sunny? Is it cloudy? Is it going to rain? Depending on the outcome of all these variables, you make decisions to say whether I'm going to play or not. If it's sunny but it's very humid, you might say, no, I don't want to play. Right? So you use multiple variables in your decision-making process to go from all the variables you have available to you to what you want to do. That's called a decision tree. It's called a tree because there's a root and there are branches and you have leaves. So you have branches and then you have leaves in the end. Topologically, this is called a tree. No other reason. And to make matters even more interesting, let's say I have 5 friends and each of us independently decides and says, okay, we're all going to vote on whether we want to play. So I might say, I don't want to play. Sumdata might say, I want to play. You might say, I want to play. And we'll just say, okay, you know what, 5 friends, let's take a majority vote. Whichever option works for 3 people, we do that. That's called, it's called an ensemble or a combination of trees. I'm one tree, she's one tree, she's one tree, and we just combine the output of all the trees to obtain a single output. Now, in real life, what's a combination of trees called? A forest. That's why we call it a random forest. Nothing fancy. These words are very intuitive. So this model that we're using is called a random forest because I take outputs from each tree, I combine, I vote them, and I basically say what's happening. So I use this model on my data, and I come up with ways to be able to make predictions. Now, one thing I want to tell you about machine learning, which I just want, if you want to carry it away with you, is the following lesson. There is a very big, have any of you heard the word hallucination in the context of generative AI, which is the current rage? When they talk about GPT, they talk about hallucination. How many of you have not heard of it? Okay, hallucination is making up stuff. That's not true. The engine is forced to give you an answer, so it'll just make up an answer. That's dangerous, right? Because you can't trust it. So the same thing happens is, if I was to tell you, how many of you have not read Harry Potter? I haven't read Harry Potter, so right, okay. How many of you have not read Harry Potter books? Yeah, I haven't read it either, so I only watched the movie. So yeah, okay. If anybody was to say, predict what's going to happen in book number 8 based on books 1 through 7, that's a hard problem, right? You can make up theories of what will happen in book number 8, right? But the possibility space is so huge that extrapolation is not going to work, right? You could be at every storyline is like a tree. Something can happen with one character and go all the way, or nothing might happen. Something might happen with a different character and go all the way. A lot of choices, right? So the same thing happens in the case of machine learning. There's something called overfitting. Overfitting basically means memorization. I'm very good at the stuff that you ask me to memorize, but the moment you ask me to work on a new problem, I fail. That's called overfitting. So beware of machine learning algorithms that have great performance, because normally in most places, they won't tell you the performance on the training data versus validation data. Just because I did all my homework problems right it is a better guarantee of doing well on the exam, but there's not necessarily a correlation, right? So it improves your chance of success, but that's why you do a test. The test is supposed to be a place for you to understand how well you understand the material, right? So unless you can do that's why we take tests so seriously, because it's an objective evaluation of how well you know the material. The same thing needs to be done for machine learning models. And machine learning models, they obfuscate all this stuff in a box, and they won't tell you what is what. So again, if you're going to be using AI, be very clear to ask, is the performance that you're reporting to me on a validation set or on the training set? The training set is like homework. Validation set is like what you get in the exam. I want your exam scores. I don't want your homework scores, right? So that kind of thing. So we build this model, and the model does fairly well. It does about 80% accuracy, which again, don't read too much in numbers. It's reasonable. It's better than 50%. And now when I do that, I run the model on all the remaining data. Remember, I had 22,000 images. I only have 33 examples to learn from. So now I let this algorithm loose on all the data and ask which genes or which drugs are causing the effect of going from this to this. Okay. Turns out there are 51 genes which, when knocked down, the cool thing about biology now is you can do essentially genome surgery. You can knock down every single gene in the genome through techniques like RNA interference or CRISPR-Cas. CRISPR-Cas got a Nobel Prize a couple of years ago, right? Jennifer Doudna got a Nobel Prize for CRISPR-Cas. It's a way to be able to silence a gene, right? So you can take every gene in the genome and silence it independently. That's what you can do. So imagine now, if I told you that 51 genes in the genome that, when knocked down individually, can cause the change of the phenotype from mesenchymal to epithelial. That gives you 51 possibilities to work with. But it's still too much, because when I went to my clinician colleague and said I have 51 genes, he said, I can't do this many experiments. Reduce it even further. I said, okay, here's what we're going to do. I'm going to skip this. And I said, let's do what Netflix does. Okay. And we said, well, I showed you 33 movies and you picked 33 that you liked or didn't like. Let me give you some more movies to watch and you tell me what category they fall into. So he spent another afternoon, and here's what I did. I had built a model right before. I let that model do on all my data, and I asked the question, on what data is my model most confused about? That's how we learn as students, right? What do we do as students? There is stuff that you know, and there's stuff that you know you don't know. And there's stuff that you don't know that you don't. Right? The first category, you skip through that stuff very fast when you're doing revision for exams. The second two, you need a little bit more practice. Right? And that's why you have teachers. You go and say, I don't even know that I don't know this. Can you tell me what I'm missing? That's how we learn from our teachers. It's exactly how we train this model. We make the model to be the student, we make the human to be the teacher, and we say the model is going to ask, the AI model is going to ask the teacher and say, teach me what I'm missing. So now I get some very specifically targeted examples. So it's like your teacher is ticking problems for you to review, to say, study these problems. You don't have to read the whole book. Just study these problems and you're prepared for the exam. Exactly what's going on. This guy picks the problems that the model needs to learn so that the exam can go well. We do that, which is again something you've all seen. I told you already. This is what happens in practically every collaborative platform. Which friends to make, which books to buy, what music to listen, what movies to watch. So we do that. The second iteration, we build a model. This iterative model is trying to annotate with a human, clustering, classification, keep going down this loop. Again, don't worry about the mechanics. I hope the intuition is clear, right? And then we do it again. I'm run the model again on all the data and see what happens now. I reduce my genes from 351 to 256. So I brought it down by about 95 genes. I can keep doing this until I come to a point that is much more manageable. So far, so good. Okay. Now, that's all very nice, Arvind. You've shown us some very cool, mathematics and some modeling. What happens to the cells? Show me what happens to the cells. So here's what happens. We take these drugs, and here's what we did. So this is triple negative breast cancer for people that are interested. Very aggressive form of cancer. We want to find drugs. That's the story in this slide. And we came to this table. The whole upshot, all that we did converges to this table. We built two systems. Like I told you, 2D culture is not very good. It's okay for people mostly do, but cancer biology happens in 3D. So we need both a 3D system and a 2D system. And we asked the question, how many, after adding the drugs, how many stayed long projections? How many became no projections? And I also said, because it's important to ask the question, how many of these drugs killed the cells? How many of these drugs kept the cells alive? So there is 45 drugs or 45 genes that keep the cells alive, but cause this to happen. They cause the change from this aggressive morphology to a much more benign morphology. Okay, so we are going with this 45. To somebody else's point, cyanide will do this, right? It'll kill the cells. But unless you know how to target cyanide exactly, you can create some big problem. So let's not go there. Let's talk about drugs that are in this bucket of 45. It turns out, remember, I have used no biology in this entire process. It's images, getting features from the images, building a model, and then trying to make sense of the model. There is no biology knowledge that has been injected into this algorithm. Yet, it picks a very canonical pathway as being implicated in the transition from the mesenchymal to the epithelial. It's called the PI3 kinase pathway. The PI3 kinase pathway, for people that are interested, is a canonical pathway that's extremely druggable and is also extremely important to epithelial to mesenchymal transition in most cancers, including breast cancer. So here's what happened. We picked two cell lines. Cell lines are approximations to real cancer. So we picked two cell lines. One is called MDA MB231. One is called SUM149. These are both triple negative breast cancer cells. And here's what happens. The upshot is the following. When you don't add any drug, this is how these cells look. Okay. They tend to have this sort of these finger-like protrusions, right? You can see them. They are here, right? So that's what happens when these cells grow with no control. When you add one of these drugs, it causes some change, but not much. When you add, like there are two drugs, this is an AstraZeneca drug, this is another drug. You add these drugs individually, it does some things, but not effectively. When you add them both, there's a much bigger reduction. Now, if you're looking carefully, you can ask me, Arvind, I still see projections here. So I'm not saying there's going to be complete arrest. That's real biology for you, right? It's not night and day, right? It'll be like not black and white, it'll be like gray. So you got to now keep iterating on it to get to a point where you find combinations of drugs. One of your friends brought it up, right? If you use a combination of drugs, this combination of drugs, PI3 kinase inhibitor and some other drug, they come together and cause a significant reduction of the mesenchymal behavior from typical cells to when they are treated. So this entire thing was done, I want to repeat, was done with no biological knowledge, right? The model does this. The model couldn't have cared if it was books, movies, or Facebook friends. I just use it for biology. That's all. So I'll close by saying that this is work that wouldn't have happened without my clinical colleague Naoto Ueno, who's now in Hawaii, heading the cancer center there. Two very bright undergraduates, Rachel and Priyanka, and then also my collaborators who gave me this data. A lot of this stuff was taught to me organically. I didn't know this stuff going in. They taught me, and we had to keep iterating multiple times. That's science for you, right? That doesn't happen. It's not like we know what we want to do. Sometimes we have to make mistakes. We get corrected, and we keep iteratively going from there. In the last few minutes, I'm going to take a few more minutes. I hope that's okay. I wanted to, because I was talking to all of you, I was thinking if I was in the audience and I had a chance to speak to myself from so many years ago, what would I say to myself to correct some misperceptions? The first would be like I told you, biology. Never underestimate the interestingness and beauty of a subject. Give it a second chance, maybe sometimes a third chance, right? Subjects are really beautiful, and don't let the books that you are being prescribed influence you adversely. I've now seen the other side of how books are prepared, and I've realized they have to prepare books with a lot of other responsibilities. Unfortunately, it's not possible to meet all the objectives together. But just because a book looked bad when you took it in school doesn't mean that's what the subject is. It's not a dry subject, so don't do that. The second is that I don't think of physics, biology, mathematics, chemistry as different subjects anymore. I actually like to think about science, right? It's almost like we study science in early school, then when we go to higher schools, higher classes, we break it into math, physics, chemistry. That's good, but you realize that it's all still science. So these boundaries are very, very fuzzy. And you might have heard of subjects like biochemistry, which is using chemistry to do biology, biophysics using physics to do biology, right? Chemical biology, which is using looking at chemistry and biology together to be able to build engineering gadgets. And last but not the least, where I come from is computational biology. We just use computation to do biology. Just like you use pipettes, you can use computation. Right? So there's a lot of interesting problems there. It's very quantitative. For people that care about things like AI, quantum computing, robotics, almost all these topics have an immediate influence on biology today. A paper came out in 2023 where a gentleman in Carnegie Mellon University, he connected ChatGPT to a robot and let it run on its own, right? He just walked away. And a week later, the system discovered a completely new antibiotic. Completely new. It's never been characterized by humans before. That's the power of chaining AI and robotics for biology. So if you think biology is not quantitative, think again. If you like engineering, biology is a great place for you to go. I already told you this, but science is very non-linear. Very rarely do we talk about how messy science really is. So when you see an experiment like Millikan's oil drop experiment or Young's double slit or whatever it is, Newton's experiment, right? Newton's sitting under a tree, whatever it is, right? It's not as simple as it looks, right? A lot of history has happened before that aha moment, right? So if you appreciate the fact that science is a very tedious process, it's fun, but it can be tedious. then you will probably understand that we all can do science. Science is only about asking why and how. That's all it is. We all use different tools, but I think we're all scientists from the day you talk to your younger brother, younger sister, right? Talk to your little kids, right? What's the most common question? Why? Do this, why? Don't do this, why? Why is very common. Why is what makes us scientists, right? So if you can just think about doing it rigorously, you're in a good place already. One good thing I always hear about is, I think science is not a noun, it's a verb. It's doing science, right? And what that means is there's a place for everybody. What I also want to say is, let's say you're an excellent communicator. You love telling stories. There's a place for you in science, a very important place, in fact. Science communication is critical. If you don't explain the science that you're doing, what's the use? Nobody knows how important it is. Let's say you're very good at drawing, at creative, visualization and computer graphics. I cannot tell you how critical it is to be able to make good visual depictions of scientific projects. In fact, I was at a meeting in London recently where a science communication award was given to a young lady who did medical illustration. Her entire job was making illustrations of medical concepts. And she got the topmost prize for science communication from the Royal College of Pathology. That's how important it is. So don't think that science is only one way. The unfortunate problem is that people think science means some gray-haired person standing over a bench with a pipette. That's one type. There's many kinds of science. Okay, so I'll leave you with that. And if I were to, I always think about what I wish I had been told. I wish I had been told to say I don't know more often. Right? This is a very august audience. I don't want to, use an inappropriate word, but we sometimes don't want to admit that we don't know. We make up stuff. We hallucinate. Right? It's better to say I don't know and learn than to make it up because we don't want to look silly. Right? So I think accepting that you may not know everything is a very important concept for a scientist. And I want to also say people confuse being a scientist with necessarily being very intelligent. Intelligence is an important characteristic, yes, but I think hard work and persistence is even more important. Right? You can be taught knowledge-wise, but nobody can teach you how to have a better grit. Grit has to be from inside. So you have to learn to be able to say, it's okay, I don't know, I'm going to persist. And I've seen so many people who are brilliant, who don't survive in science because they don't have the grit. Grit is being able to say, well, my experiment failed, too bad, I'm going to try again tomorrow. That's the difference between people that make it in science. And there's a lot of good books. Some of them are even on YouTube. If you want, you can talk about this. And I'll also say that there's no one perfect kind of learner. Some people do well with ICSE books. Some people do well with CBSE books. Some people do well with IB. Right? They all have different learning styles. But I think there's this strength in diversity of learning. I think we should all appreciate the fact that each of us can have a very different slant to learning about science. I'll close with a report that I want to read out to you. It's very bad print, but it says this. It's a report about someone. This is not Einstein. This is not Edison. And you already know. You've already heard the stories. Einstein, was kicked out of school. Edison was called addled and all that stuff. So that's all known. This is another person. This is this report when he was in primary school, 1949. Says this is the midterm report, and it says it has been a disastrous half. His work has been far from satisfactory. His prepared stuff has been badly learned, and several of his test pieces have been thrown over. One of such pieces of prepared work scored 2 marks out of possible 50. His other work has been equally bad, and several times he has been in trouble because he will not listen, but will insist on doing his work in his own way. I believe he has ideas about becoming a scientist on his. Thank you. I can't even read anything. Well done, amazing. Yes, it's quite ridiculous. If he can't learn simple biological facts, he would have no chance of doing the work of a specialist, and it would be sheer waste of time, both on his part and of those who would dare to teach him, care to teach him. Okay, so this is the report. This guy won Nobel Prize. Okay, so the point I'm trying to make to you is, it's not like, there are always people that stand out. That's fine. What I'm telling you is that this guy almost didn't get into science because of these reports. He went to Oxford to study history or something, and by some accident, he went back to science. Like he had to graduate in a hurry or something, so he took that class, and next thing you know, he went on in that direction, right? So I'm just telling you is that, don't sell yourself short. And the good part about it is, unlike when I was in your age group, now there's a lot of open data sets available for you to be able to try your own curiosity. So I'll pass on some exercises later, but that will let you explore data on your own. But there are some cancer data sets that are available online. There's image processing available online. If you know Python or some kind of programming language, there's a lot of stuff that you can play with in your free time. So if you're interested and inquisitive, make sure that you go in these directions. Cutting it back to what I do. So bottom line, this is what I did many years ago. What I do right now is understanding tumor ecosystems. So as I told you, tumors are very, very special because they have lots of different cell types, different species all collaborating. And so what I spend my time doing right now with my students mostly, is figuring out the architecture of tumors using spatial biology. So I'm going to close with a question for all of you. There has to be a question in the end, right? So based on what I told you, which type of cancer cell is the most dangerous? Okay, but you didn't even look at my third option. Quiescent cell. What's a quiescent cell? Anybody knows the quiescent cell is? Say a little bit louder. Okay, yeah. So something like, so it's basically it's arrested. It's arrested, but it's not doing anything just yet. But all it requires is a small push, and it'll go in a bad stage, right? So that's quiescent. But I have two other answers. How would your answer change if I put these additional two options on the table? Okay, that's one option. So I would go with either 4 or 5. There's no right answer, right? But I would go with 4 or 5 because depending on where this cell type exists and what is happening in its environment, the answers can be different. There's no absolute answer. That's the beauty of biology. We don't have Newton's laws of biology. We know about evolution. We know about survival of the fittest, those kinds of concepts, but we don't have a law of biology just yet. So you can see why it's so important to work on biology. And the best part about this last thing is it's an iterative experience, right? You do an experiment, you learn something, you redefine your hypothesis. That's the idea. Okay, so I'm going to close, but I hope Dr. Rayasam and Dr. Sridhar are here as well. But special thanks to Dr. Karak, Dr. Rayasam and Dr. Sridhar for inviting me. I hope this has been useful to all of you. I'll hang around here for a few extra minutes if you want to talk. But I want to point out this is a team endeavor. I'm a very small player in this whole team. The bulk of the work is done by my students. They are awesome and fantastic, and have wonderful teachers in terms of collaborators who teach me what mistakes not to make. I make a lot of mistakes, so they are always there to tell me, Arvind, this is wrong, in a very nice way. And then, of course, money matters. Money is what supports our research. So very grateful to all these funding agencies supporting our work. With that, I'll close. I'm happy to take any questions. Sorry for going over. Okay, great. Any questions?
Useful for people listening on it.
So you said that around 256 genes are responsible for the transformation of from mesenchymal to epithelial.
Yes.
So what if instead of transforming them, we just completely remove those genome from the cancer cells?
So the problem is that genes are like very complex circuits. One gene doesn't only do one thing; it does many things. So it's like there's a dependence. If you take away one brick in the wall, you take too many bricks out of the wall, what will happen to the wall? Come crumbling down, right? So that's the problem. Is there a systematic dependence between? So what you have to do is you have to these drugs are very good at like volume modulating the gene's expression to a point where it's minimized but not completely knocked out. You could do, yeah.
Sir, I have another question.
Yeah.
Sir, if we cannot remove that certain type of gene, what if we reverse its growth? Instead of letting it grow, we stunt its growth. Maybe there is some type of, for example, for every chemical reaction, there is a reagent which speeds up the process of that reaction.
Right.
So what if we use something related to that to stunt the growth of it instead of stopping it or removing it?
I think it's a great avenue. I haven't thought about it personally, but I think there's your research project for your PhD. You picked your PhD project. That's great. But you're talking about exactly this, right? You're doing a chemistry concept to be able to think of biology and talking about speeding and reducing the speed of certain phenomena. And the best part is that I'm beginning to I cannot give this example anymore, I realized. How many of you have seen a old style radio? Okay, good. Okay, good. That's becoming harder and harder to explain. But there's a lot of parts inside, right? If you take one little valve out, a transistor out, the radio won't work, right? The same thing is happening here. Is what we do is you have all these genes are like different parts, and there's dependence. And if you want to talk about, you need like a physics perspective, a stoichiometric perspective to understand in what combination and in what order to reduce these genes to be able to get the desired behavior. It's almost like changing stations, right? FM, whatever, FM1 to FM2 or whatever. Thank you for understanding that concept. This is a hard question. Yeah, go ahead.
Sir, there are many researches that are taking place in the field of reducing cancer. However, there is not much information of what is causing cancer. Because if we look at the statistics of it,
yes,
cancer has increased exponentially in the recent years.
Right.
So is there, because these data that you're talking about, they can be achieved after cancer has started growing and we can quantitatively measure it. Is there a way in which we can find out using data itself that what exactly is causing cancer these days more in this generation or in this time frame?
Okay. I'm smiling because I'm very proud of your question. I think exactly the kind of question we need to ask. So what you're asking is a question of epidemiology, which is lifestyle, exposure, food, demographics. What is the combination of these factors in conjunction with our DNA that is either hastening the process of cancer formation or doing something unique? Like if you talk to any of the old timers, right? Like grandparents, we used to eat natural food, right? Everything, no, what is, I mean, I'm sure you all heard this, right? What is all this pizza burger business, right? You all want to eat fresh food, right? And they had a certain lifestyle, they had a certain kind of exposures to different things, and that had a consequence on their life. The same question comes back to, and your question is very pertinent because there is now a silent bomb that's happening. It's called early onset colorectal cancer. For the weirdest, we don't understand why. But people below 40, there's a big spike in young people getting colorectal cancer. Nobody knows why. We have better medicines today. We have better products available, yet young people are getting colorectal cancer. Why? So you're asking a question about if we were to understand the variables that cause this kind of biological behavior of addition of cancer becoming a bigger problem, what can we do at the policy level to be able to control for those kinds of problems? That's a very deep and serious problem, right? Because on one hand, you have to study the statistics, connect that to epidemiological data, which is hard to get because you have to basically go and do surveys, right? Get surveys like, okay, what did you eat today? What did you eat yesterday? What's your age? Where did you grow up? All those variables. Connect that and build models like I talked about, that connect that to cancer incidence. And then ask the question, well, if it turns that most people that got cancer in this area live very close to this factory where they dump a lot of chemicals into the water. Okay, now we can say to the government, maybe do something about that. But not most things are not that black and white. It's about the food we eat. It's about the exposures that we have. So I think your question is extremely important. It's the right question to ask. The second PhD problem. Actually, it's not just one. It's at least 3 PhD problems right there. Good. Great questions. Keep them coming. Any other questions? There is no bad question, by the way. Every question is good. Go ahead.
Sir.
Yeah. Go ahead.
So nowadays, most of the people, many people are talking about reversing life process.
Okay.
Like, what if instead of letting the cell age, we reverse it somehow to make it look in? So is this possible in cancer also? What if we, instead of, so for example, if we have to enter this auditorium, we have to climb the stairs and turn right. What if instead of letting the cell turn right, we make it turn left?
Great question. So have you heard of telomeres?
Sir?
Have you heard of something called telomeres in the genome? Okay. So they are basically the tail ends of the chromosome, right? And what happens is that as we age, the telomeres get shorter. So there's a whole field of telomere therapy where they figure out ways to make sure that the cell is aging at the right time. What cancer cells have learned to do is they've learned two things: how to survive despite having a bad genome, and second is how to avoid being detected or cleared out by the immune system. They almost figure out ways to make friends with the immune system to say, you know what, nah, I'm not that bad of a threat. I'm just going to hang around. Right? I'm not really bad. And then slowly but surely convert the immune system to help it. So what you're talking about is it's what is called a systems biology problem, in the sense that it's very hard to just change one thing. You have to change a bunch of things. When a weed grows in your garden, there are two ways to do it, right? You just add a weedicide and kill the weed, or you try to nourish the soil differently. The same thing happens in cancer. Can we create a better environment for the cells to take different choices in their growth pattern? But again, what you're saying is definitely something that is being worked on very actively. You look for ALT, look for telomere therapy, those kinds of things. You will find a lot of things. And there's again also this concept of stem cells. So if you've heard of stem cells, you know that stem cells are very early cells that differentiate into different categories. There's also a stem cell for cancer. It's called cancer stem cells. So the question is, if you get a cancer stem cell and you kill the cancer stem cell at the very beginning, it could never progress into more serious forms of disease. It's exactly the kind of things you need to think about. Very active area of research. Good. Any other questions? For single gene diseases, CRISPR has been shown to be quite promising, like for sickle cell disease, those.
96% efficiency it has shown. But even for lung cancer treatment, the CRISPR has been the most advanced treatment for lung cancer and everything. Lymphocytes are taken out. And then the genes are suppressed using CRISPR, and then they are transferred back. So how do you compare the limitations of CRISPR with drug treatment using AI?
How do you compare the limitations of CRISPR compared to
drug treatment processes?
So I think CRISPR gives you a level of precision that has been so far not possible to achieve. So now you can knock things down very selectively. The question is still, the problem is because it's a combinatorial system with so many moving parts, which ones to hit that will cause the optimal outcome? That's the problem. And I think AI is trying to get to problems in that space. It's a hard problem because it's like saying you have all these networks, all these nodes in a network. Which nodes are you going to hit to get the maximum impact? Right? That's still a problem that is being actively worked on by computer scientists. But it's again a place where AI could meet biology. Yes.
There are the disadvantages of
Sorry. Yeah.
There are the disadvantages with the AI too. It is not always the efficient way, and it's not. We cannot also go with the manual. We need a balance of it.
Right.
So when we are introducing onto the sensitive sides of dealing with the biology or the miniature parts, how efficient could the AI be?
But again, I think I think we need to rethink what we like to call AI, right? If AI is just this notion that you will learn with enough data and you can model relationships with what you have computationally, and that's a very limiting thing. I think the part where AI ought to go is what is called scientific AI, where you need to sort of introduce AI as one more reagent
And then
in the scientific field.
there comes my another question related to same thing. As the policy making keeps on going, there are different parts of the elements where the society want to focus on, be it like economics, the part of science and everything. But when it comes to science, dealing with the creative side, just like we have gone with the computational biology where mathematics and biology plays a role, and there comes like we are narrating it down as well as we are converging the whole part of sciences together, and still there is a competition for the creative sciences. So we are going with the research, we are trying to go with the funding, and then why do we have to go with the competition for the fundings?
For funding?
Yeah, for funding or to get a kind of support to go on with the research and then make it to a better success rates.
Yeah, I think your question is a deep question, but I'll try to answer it in as short a manner as I can, which is that I think what competition creates is a sense of priority for certain aspects of what needs to be investigated first. Like you can imagine, there are foundational building blocks to every science. And unless the foundational blocks have been fully resolved, building upon those blocks becomes very difficult. So if you're talking about doing something that is much later in the game, then arguably one would say, well, let the foundations first be fully resolved, and then we can fund the second wave of the incremental research. So I think in some sense, competition is good because it helps to create a consensus around what problems are of immediate priority versus of later priority. That is the only reason resources are always limited, both in terms of time or money. So one has to be able to prioritize. That's the mechanism that I understand.
Is there anything other than that we could deal, like instead of money, there are so many other resources we can take over and then make sure we do have a success rate with the research that we are doing? Or
yeah, so there is philanthropic science, there is foundations, there is a lot of people that are interested to use crowdsourcing and crowdfunding to be able to do science. They're all possibilities. Yeah.
So, one suggestion. Like as you said, in order to understand science, it is very important to integrate different branches of science, like physics with biology, chemistry, and etc. But once a student is in a graduation field in engineering, will not have a biology background. But like people like us who are already into this field of science, like we are doing our graduation in biotechnology with biochemistry, and we do not have background in physics related to data mining or AI or something. So like you had a crash course in biology, as you already told. So in order like we integrate our subject with data mining and etc., what are the few courses which you would suggest us to also complete during our bachelor's degree?
Yeah, so I can't speak to it very broadly because but I know one thing for certain. I know that there are programs CSIR, for example, conducts. They're like boot camps for like 1 week or 2 weeks, right? Where the idea is to be able to say, a biologist with no programming background, how do you learn how to write your first program for analyzing data? Similarly, the other way around, right? You're a programmer, you have a lot of good software background, but you don't know biology. Come, let's teach you biology for the next 1 week and talk about problems and data in that space. And that's definitely one of the goals here, is to use this as a nucleus, but then to build on it by saying, here are some experiential materials that we can give you that will let you iterate and learn whatever you think is missing in your training. But I think the time is great right now. A lot of institutions in India offer courses like this, including in the summer and those kinds of things. So I think,
like, what are the courses which you would suggest? Like, in order
I think it's very background dependent. I would say that, I think if you're a pure biology person who's never done programming, getting one programming language will help you, because you need that to be able to talk to your data. Similarly, statistics, right? I think people don't talk about it enough. There's a thing like doing bad statistics, because you can get wrong results. So I feel that,
like, if you have seen like computer aided drug designing, CAD and everything, they speak about Python and everything, but a biologist will never understand what actually Python is being used in CAD, but they only know how to use Python in that particular procedure of using CAD. So instead, if you can understand Python language and everything, that will also help you go in depth, I feel.
Absolutely. So I would say that's a good point to bring up. With the resources available on the internet now, with Coursera, YouTube, I don't think there's a need to get permission anymore to learn anything on that you want to learn. So there's actually a very nice 10-hour course on Python programming on YouTube that is really, really good. My students take it all the time. It's free, right? I can take it, you can take it in the weekends, you can take it in the evenings, whatever it is. So I think the idea is just to be able to just find resources that you think will help address whatever blind spots you personally might have in your process of discovery. There's no need to wait for some magical program to come out, because I think we can all search for these things now on our own.
Also wants to add.
Oh, yeah.
Yeah, am I audible?
Yes.
Yeah, so great talk, Arvind. I really enjoyed it. And I think as a biologist, even for me, it was a lot of learning. And I really like this random forest. I've heard about it, and today I really understood why it's called random forest. And I think I just wanted to thank you actually, because I think it's not only for the students, it's good for all biologists. And I really like the concept that it is good to say, I don't know. And I think it's really encouraging for the students. And I think we have the next project already. The last question, the student, she talked about, for biologists, how do you teach programming or big data? With your help, we could think of, coming up with some workshop. It could be an online workshop for those students, biology students who want to learn more about data. I think she makes very good point that Python, people know the tools, but how to utilize these tools, how to ask the questions. We forget always that these are only tools, be it AI, be it machine learning. It is what is the question you are asking? Like you talked about cancer. There is only value to the AI if it's really solving something. Right. So I think we could think of some focused workshop where we really expose some of the students who are interested from biology to look at, because you are coming from the perspective of engineering to biology, it should be other way around also with respect to the AI. So I think we could do something. So I just wanted to thank you and Somdatta for organizing this, and we look forward to working with you again.
Thank you, Dr. S. Was there a question? I thought. Any question? Sure.
There was just this one last thing that I wanted to say. You were talking about taking surveys and combining it with data. Instead of that, just like you had performed in your research, if we use, or maybe before that even, if we use different combinations, like for example, in the recent years, plastic has been increased in our lifestyle. Everything, like right from milk to plates, everything is in plastic. So if we use those cells that you talked about, and we try to add like different components of our lifestyle, like maybe plastic, and then maybe this particular gluten rich nutrition, and then we try to see how the cells are behaving, that might tell us why these cells are getting so aggressive recently.
I think it's a beautiful idea. It's a great concept. There's actually a whole area called three-dimensional bioprinting, where you can use the ink can be of any type. It can be cells, it can be plastics, it can be different chemicals, and you can create any kind of configuration spatially of these different subspecies to create a ecosystem of these different pieces. Nobody knows what's the right way to do it, but technologies to do it are just coming online. So if you got into this right now, by the time you get to your PhD, you will have a very good set of topics to be able to create something very important. I think you're asking a very deep and very important question.
So I have a question. So what if instead of focusing on genomes that are causing cancer or transforming cancer, what if instead we focus on the genomes present in our body which oppose cancer? Can we strengthen it or make it more potential to oppose cancer more powerfully?
Excellent. So the heart is an organ that doesn't get that much cancer. Turns out it's one of those organs that doesn't get cancer as much. So that's one thing. There's also something called the Peter's paradox, which is that there are certain organisms that don't get cancer at all. Right? It's very surprising. Their cells are dividing, they're doing all these things, yet they don't get cancer. How is that possible? Can we learn from their genes and figure out how to bring those behaviors back to our genes? So learning is, I think what you're talking about is picking example. And again, same thing we do even in clinical science. And this was done actually in the beginning of the COVID era, was that the people never got COVID. They were exposed to it all the time. They never got COVID. So then the initial part was, before they had the vaccine, they said, can we just get the antibodies from those people and make antibody therapy for COVID? So that it's called exceptional response, exceptional, they have some exceptional property in their body that makes them immune to certain diseases. So can we use those and then engineer them to be able to learn what their bodies are doing right, that we could then replicate in other people's systems as well. So I think learning from biology in these ways is very interesting. Absolutely. Good. Are you all in school? What grade are you in? I wish I was like 1/10 of your abilities when I was in. Oh, AI, excellent. Excellent. It's been amazing. I'm really glad to have spoken to all of you. But yeah, if there are any questions that I can answer even offline, I'm happy to do it. Great questions. If you're inquisitive at this level, science is going to be in great hands. I'm very, very hopeful. Thank you. Thank you.
Thank you so much, Arvind, for this excellent talk. Clearly, it was extremely engaging for all of you who are here with us in the CCMB lecture hall today. And I hope that the talk was equally exciting for people who were listening to us online, not only on Teams, but also on YouTube. Yeah. So I would just like to close this session, thanking Arvind for this excellent session, excellent inauguration to the series, and look forward to meeting you all next week again, as Dr. Lipi Tukral will be giving the next talk, and she will be talking about how she uses big data to understand protein structures. And so that will be next Friday, 12th of April, 10:30 onwards. So that's going to be how the Futuristic Friday series is going to be designed as. Thank you all very much. See you again next week.